So yeah, and thank you to all the speakers that went before me. So what I thought I would do today is give you a one person's perspective, and it's a computational perspective on what we think is coming. I think we all think this is coming, the biology revolution. And in thinking about this, we can ask ourselves, what insights allowed computer science to have these remarkable impact on our life and to drive the information technology revolution? And I think three things could be pulled out. One is this profound distinction between hardware and software, and a focus on the control of information, not just energy and matter. And in particular, the development of these things we call compilers, which allow you to abstract away from low-level details and think about the rational design of modular functionality. And so this is sort of the progress in the field where we used to program by literally moving wires around. So you had to think about the hardware. The hardware was everything. And then eventually we got this nice machine code, still very much wedded to the hardware. But eventually we have these modular sort of object-oriented languages where if you're writing things like this, you really don't need to know very much at all about what's underneath. And the reason I think this is relevant is because in biology, we're getting good, as you saw today, we're getting really good at manipulating molecules and cells, the hardware. We're getting much better at this. But we're really a long way from control of a large-scale form and function, such as, let's say, regenerating an arm. And so the question is, can we work to move biology beyond a focus on the machine code? And I'll just give you two simple examples. We know a lot about DNA, but here are two puzzles. We have these planaria, of which you'll see more in a minute. But basically, these guys reproduce by, these are flatworms, they reproduce by tearing themselves in half and then regenerating. Okay. So over the last, let's say, 500 million years, these guys have been practicing somatic inheritance. Every mutation that doesn't kill a neoblast or a stem cell makes it into the next population. Their genomes are a mess because they accumulate changes continuously. And yet, despite all of this, they are perhaps the champions of regeneration. Every time they regenerate, their anatomy is spot on perfect. Okay, so we have this puzzle, right? What is the relationship between this amazing patterning ability and their genome? Which is, I mean, these animals are mixoploid. All the cells have different numbers of copies of the genome. It's crazy. And you can ask yourself another question. If we take two species of planaria, let's say with a flat head and a round head, and you mix the stem cells from one into the other so that there's now a combination, and you cut this off, what shape head are you going to get? And we have no idea. And the reason we have no idea is that while we understand a lot about the molecules and the mechanisms that are required for some of these things, we do not understand the algorithms that link large-scale shape and pattern to the underlying hardware. And so the reason this is important is because if you look at some of the major problems that biomedicine has, so birth defects, traumatic injuries, cancer, aging, what do all of these things have in common? Well, one of the things they have in common is control of anatomical structure. Errors of initial patterning, loss or damage in adulthood, defection of cells from the anatomical plan, aging, and so on. And so the idea might be that if we could address not only the mechanisms, but the algorithms of patterning, such that we had control of anatomical growth and remodeling, we could really make an impact on many areas of biomedicine. So the first thing I want to talk to you about now is this amazing ability of at least some types of bodies to edit their own anatomy. So we all start life as a single fertilized egg cell. This thing reliably self-assembles into one of these remarkable morphologies. But keep in mind, stem cell differentiation is crucial, but it's not enough. This thing is a teratoma. It has bone and teeth and hair and various other structures. But of course, it's not an organism because it's lacking the three-dimensional structure. So this is development. Development itself is, however, is not hard-coded or hardwired. So we know that we can split or actually join embryos of some species, and what you get are perfectly normal, in this case, monozygotic twins or slightly larger mice. So this is a system that can make up for pretty drastic early deformations. During adulthood, here are these planaria. They can regenerate from any kind of cut. So any cut you make, the fragments will restore exactly what's missing. And even vertebrates, such as this axolotl that you've already heard about, can regenerate many of its organs, its limbs, its eyes, its jaws, and so on. So this is regeneration is an amazing process that repairs unpredictable perturbations and then stops at exactly the right moment. And that actually is a really important issue, is how does it stop? So regeneration is not just for so-called lower animals. Even the Greeks knew that the human liver was regenerative. We don't know how they knew that, by the way. Deer, we know, regenerate centimeters every day of new bone and skin when they're regrowing. Even human children below about the age of 11 can regenerate their fingertips, actually. And this is the kind of example that challenges us to think about information flow in this system. If you transplant, these are really old experiments, if you transplant a tail to the site of a limb over some amount of time, even the cells at the tip of this tail get the message that this whole structure needs to be something different at this anatomical location, and they slowly remodel, right? So there's this massive sort of anatomical surveillance and readjustment. This is the work of Doug Blackiston, a postdoc in our group, where he actually showed that this plasticity is not only structural, but it's also functional. So if the primary eyes are missing, but you put an ectopic eye on the back of this tadpole, you can use a machine like this that we built to train these animals to visual cues, and these animals actually have no problem seeing out of that eye. They can see perfectly well. So the brain adjusts its behavioral programs to a visual input that now comes from this weird patch of tissue on its back, as opposed to where it could have expected to get it from for millions of years of frog evolution. So an amazing degree of plasticity. And this is another example of how we have to understand how these things process information. This is the work of Danny Adams and Laura Vandenberg in our center, where normal tadpoles remodel their faces and become frogs, but so do Picasso-like tadpoles. So if you make a tadpole where all the organs are in the wrong place, so the eyes are shifted, the nostrils are all over the place, okay? So you do that, and then these guys will just as well remodel, move their organs around, and eventually you get a pretty normal frog. So as a computer scientist, you look at this and you say, okay, well, fundamentally, this is a computational problem. These guys have to know what shape it's supposed to have. They need to know what shape they have now, and they need to know how to get from here to there, and then they need to know when they should stop growing. So one way to think about this is as a general property of pattern homeostasis. Development, regeneration, all of this is a kind of pattern homeostasis where the system is trying to maintain and get back to a large-scale anatomical state. So beyond this, which we're all used to thinking about as you've got your gene regulatory networks, your proteins interact in some way, and then this marvelous anatomy sort of emerges in development, we really now have to focus on this feedback loop where, yes, this is true, but then after unpredictable perturbations, this system is able to process information both in the gene regulatory circuits, but also in the physics of it. Okay. Oops. Okay, this thing. Yeah, didn't really know. This thing doesn't seem to work yet. Anybody got a pointer that works? Another one. Everything suddenly died.
So this goes into the...
Oh, I don't need that. I just need the pointer.
Oh.
Oh, there it is. Okay, thank you. How did you do this? Okay, well, okay. So yeah, so we need to focus on this information processing loop that allows tissues and organs to change position, change shape, and change gene expression as whatever's necessary until the correct shape is reestablished, and then they stop. So since that's the case, thank you. Perfect. Much better. Thanks, Junji. Yeah, excellent. Okay. So. The question is, if this is a homeostatic cycle, the most important thing about a homeostatic cycle is that it has to store the set point. Everything from your thermostat on down, if it's going to have homeostasis, it has to store a memory of its set point, in this case, a pattern memory. And if that's true, this suggests a hypothesis. Can we target this pattern memory? Can we change it and force the homeostatic process to go to a different direction? So what I'm going to tell you now about is our work on non-neuronal bioelectricity, which is one probably of several media by which bodies accomplish this task. I don't have to go through this anymore. You've heard a nice introduction where basically each cell has its own voltage gradient across its membrane, the differential of resting potentials across a field of cells such as this tadpole here form what we call a VMEM pattern, or a snapshot of bioelectric activity, and then there are a variety of bioelectrical signals that are temporal changes in this pattern. So these electric pre-patterns are in two forms. There are your normal endogenous pre-patterns, such as this electric face, first discovered by Danny Adams, which shows you where the different organs and the different gene expressions and positions of the face are going to form in this tadpole. And then you've got your pathological patterns like this, where this tumor induced by a human oncogene is revealed by its aberrant depolarization even before it's morphologically apparent. So these and then basically what we do technically is we are able to characterize largely using fluorescent dyes, but increasingly using genetically encoded reporters. We're able to see these electrical conversations as they take place, and then we're able to model them. And this is the work of Alexis Pajtak, who's here in the audience, who does a lot of this remarkable computational modeling that allows us to understand how this all works. So beyond modeling and observing, now these are our functional tools. Here's our electrical network. As you heard today, we're able to modify both the topology of this network by changing the way cells talk to each other through electrical synapses, known as gap junctions. And of course, we can go in either with light or with drugs to, or by misexpressing other channels to actually control individual voltage states of these cells. So it's corresponding to synaptic plasticity and intrinsic plasticity if you were dealing with neurons. So I'm going to show you now some examples of what we've done by targeting this communication system among cells. And so we're going to explore these examples, but I tell you that just to keep in mind, none of what I show you was done with applied electrical fields. This is all molecular pharmacology or molecular genetics. So one application of this is that we've shown that if you do put in a tumor, so let's say a KRAS or a REL3 mutation, normally in Xenopus it makes these tumors and they're marked in fluorescent with a fluorescent fusion protein. But in fact, if you do the same thing, but you manage the electrical state appropriately such that these cells do not depolarize, they keep in electrical contact with their neighbors, even though the oncogenic mutation is strongly present, in fact it's all over, but really strong here, there is no tumor. So you can actually manage the outcome of this and keep the cells sort of working towards their normal tissue plan, despite the fact that this oncogenic mutation is there. And we've done it in a number of different ways, including optogenetics. Another example of restoring pattern, and this is the work of Vannevar Bush, a postdoc in my lab. This is a normal tadpole brain. You see here forebrain, midbrain, hindbrain. If you treat this animal with teratogens, or else you introduce a mutation in a gene called Notch, which is a really important neurogenesis gene, the brain is completely disrupted. Basically you can see here total loss of structure. Forebrain is almost gone, midbrain is a mess, and so on. But the remarkable thing is that even in the presence of these teratogens, including the mutation in this critical gene called Notch, you can get largely a normal brain with proper gene expression, proper anatomy, and proper memory. They get their IQs back actually if you superimpose and enforce a brain-specific voltage pattern in early development. So the take-home message from the last two examples that I showed you is that despite, for example, mutations or various other disruptions, we can enforce correct anatomy by managing the bioelectric events towards a correct state. And so here's another example where we're working on leg regeneration. So a frog, unlike a salamander, does not regenerate its limbs. And so after amputation, 45 days later, you either get a spike or nothing. And so what we can do is apply a cocktail that adjusts the wound site to a particular bioelectrical state, turns on regenerative early genes like MSX1, and then it kickstarts the whole process of leg regeneration. Now these legs eventually look pretty good. You've got a toenail, you've got some toes, and you can see that this leg is touch sensitive and motile. The animal can move out with this leg. This whole process takes months. Initially, the treatment with this ionophore cocktail lasted exactly 1 day. So this is a kickstarting event where you can trigger the whole module of leg building by very early stimulation. And so with our collaborators in David Kaplan and Annie and people in his lab, we're working on making bioreactors and transitioning this to a rodent model to try and move limb regeneration forward. So I've shown you 3 examples now where we can try to reinforce or repair normal pattern. Now the question is, beyond the repair of normal pattern, how far can we go with this? How much control over anatomy can we get? And I tell you now, I'm going to show you some really weird creatures in a minute. None of it is Photoshopped. These are real, actual, living, beings that are in our lab. So these are all real things. So one thing we found out how to do years ago was to induce specific organ formation. So here's a tadpole. The embryo has been injected with a particular ion channel into cells that are going to give rise to a portion of the gut. And what happens is, on the gut, you get this complete eye. And this is the work of a graduate student, Sherry Au, in my lab from years ago, and also Pai as well. And these ectopic eyes have the same cell layers, retina and optic nerve and all this, as a normal eye would, but they're made out of tissues that were previously thought to be incompetent to form eye, for example, endoderm. In the case of planaria, what we can do is we can cut off the head and the tail, take this middle fragment, and alter this normal electrical gradient that tells the animal where to put the head and the tail, and thus you can get your two-headed forms or your no-headed forms. And these guys are perfectly viable, and you can ask some really interesting questions about their IQs and behaviors and things like this. But you see what's going on here. This is respecification not of specific cell types, but of whole organs. So what we didn't do was have to micromanage this process to say where the retina and all the components of the head are going to go. We're literally dictating a large-scale anatomical features. Beyond that, this is a project of an undergraduate student, Maya Emmons-Bell, who found out that what you can do is you can take this species of planaria with a triangular head, cut off the head, take the middle fragment, perturb the electrical topology of that fragment, and you can get worms with a flat head, like these P. felinas. You can get round heads, like this S. mediterraneus, or you can get the normal triangular head. And note that you get not only changes in head shape, and we've analyzed this with morphometrics and so on. So not only do you get the shape of these other species, but you actually get the brain shape and the stem cell distributions appropriate to these other species. These guys are about 150 million years distant from the planaria that you started with. And keep in mind too, there's no genome editing here. The genomic sequence is completely normal, has not been touched. There are no transgenes being introduced. This is a purely electrical modulation of the network that gets you from this species-specific morphology to a completely different species-specific morphology. You can go further than this, and we've seen that various, so apparently planaria don't even have to be flat anymore. You can take these flatworms and you can get these spiky forms, you can get little hats, and you can get these tubes. This is largely the work of Fallon Durant, a grad student in the lab, and other members of our worm team. But really, this is starting to show that a lot of aspects of the anatomy you can get from a single genome are really pretty diverse. And so I've showed you some examples of non-genomic pattern control, and so we start to wonder, okay, where is this pattern, right? To go from here, from an early set of blastomeres to this really precisely, this is a cross-section of a human torso, so to a really precisely patterned structure like this. So where does this pattern come from? And we did this experiment, so this was Nestor Oviedo and Junji Murakuma and various other people since then. And we started with this question: if you take a middle fragment and you treat it for just a couple of days, you make this two-headed worm. And then what we're going to do is we're going to take this middle fragment, weeks later, after all the reagents are gone, we're going to put it in plain water, we're going to cut it again. And so the current paradigm would say, well, the genome is still the same, and you've gotten rid of this ectopic extra head tissue, whatever, if it was an organizer or something, that's gone. You're keeping just this nice normal gut. What's going to happen? Surely you're going to have this. And the answer is no. What happens is, in perpetuity, every time you cut it, this fragment will continue to regenerate as double-headed. And then, in fact, as of recently, Fallon's work has shown how we can actually set it back. We can take these guys and set it back to being normal. So what we have here is the fact that the target morphology, the shape to which this animal regenerates upon damage, can be rewritten or edited in a permanent way. And this transient change respecifies it in a stable way. So this is really starting to push the issue of memory, because this is stable, and it's almost like a flip-flop in the sense that it can live in two permanent states. It can be like this or like that, and we can shift it back and forth. And we've started to think about this as the relationship of this kind of signaling to to brains and understanding of the nervous system. And I point out, this is one cell, and this guy is sort of hunting for his food here, but this is one cell. And I just point this out to remind us that complex behaviors and information processing were around long before brains showed up. So, brains and neurons evolved from much more simple cell types that did a lot of information processing, and you don't have to be a neuron, and you don't have to be a brain to have memory, to have flexible dynamic behavior, and to solve a lot of problems. And so, one of the most exciting recent stories, and this is also Fallon Durant's work in Biophysical Journal just from a few months ago, we have this remarkable finding that planaria, which have, so here's a flatworm, here's some gene expression showing where the anterior genes are, and so these guys, of course, make single heads, but we're able to generate animals that are anatomically normal, histologically and molecularly normal. They have the same markers, and they have the same stem cell distribution, and all these things are the same, but they're able to generate two-headed forms. How is that possible? And it turns out that the reason it's possible is because these anatomically normal worms are able to harbor a bioelectric distribution that is not normal, in fact. That is, and you don't know anything is wrong with these guys until you check their electrical pattern memory. And so the kind of amazing thing, well, there's two amazing things. One is that the same body, this is very much like memory in that the same structure can store distinct patterns inside, but the patterns are not indicative of what the anatomy is now. This bioelectrical state is not telling you what the anatomy of the animal is now, it's telling you what is going to happen if it is cut at a future time. This is an information structure that the animal is going to consult if it gets cut in the future, but in the meantime, the anatomy and the molecular histology are perfectly normal. And it's not a surprise that this all starts to sound like the brain because brains use the same kind of scheme. You've got your hardware, which is these ion channels and electrical synapses that drive electrical behavior, electrical dynamics that result in memory and cognition and behavior. So this is a movie of a zebrafish brain, where zebrafish think about whatever it is that zebrafish normally think about. But we have a similar situation here where, in fact, all cells have the same ion channels, gap junctions, and so on. And so here's this movie again, suggesting that much as people in the field of neuroscience are working on neural decoding, so this idea that you will be able to read electrical states in the brain and then infer the cognitive structure, infer the semantics of these electrical states, like this person is thinking of a particular image, we ought to be able to take readings of the electrical activity of these systems and be able to, if we understood the bioelectric code, infer what the pattern is going to be. In the case of planaria, it seems to be fairly straightforward, although even that is much more complicated than it seems. But in the end, it's going to be quite a challenge, but the idea is to decode this and maybe even use that information to help out the neuroscientists in what probably is an even harder problem. So another way to think about the role of bioelectrics in this is that in between the genome and the anatomy is this layer of physiology, these bioelectric networks that have really dynamic flows and changes of voltage, and that the state space of these electrical circuits possess attractor states, stable states that correspond to distinct anatomical outcomes. We've only found a couple, no doubt there are others that we haven't found yet, but basically when the circuit lands in each one of these different stable states, it issues commands to the different cells and the various genetic circuits that build different structures. And this is well known in neuroscience that these kind of networks, this is basically a map of individual memories in a kind of artificial neural network that could be used to store information. Each one of these things is literally a memory and this might be a good way to think about these things. So I've told you is that all cells, not just neurons, form complex bioelectrical networks, that targeting these networks allows rational control over large-scale anatomy, and we're getting better and better at doing this. That in some cases, these network states can override what would otherwise be a genome default outcome, and that activation of specific pattern memories provides modular control, where we can call up eyes, brains, limbs, and certain other organs without having to specify all the low-level details that we don't know how to specify anyway. And so what I would suggest is perhaps a change of the hardware-software metaphor, where usually what you read is that the DNA the DNA is the software and that the cell is the hardware that interprets it. But another way to think about it is that the DNA encodes the ion channels, and thus the DNA is what specifies the hardware. The software is actually all the electrical dynamics that run on the hardware that you get when a bunch of electrically connected cells are sitting next to each other. And the reason that's important is because this software can be edited separately from the hardware. You don't need to, much like you can put different algorithms and different memories in a set of chips without having to yank out the transistors, the same thing is here. You can keep your ion channels, but if you knew how to alter the electrical dynamics with, let's say, ion channel drugs, specific compounds that would knock the system into different attractor states, you might be able to control what happens without having to change the hardware too much. So in the last couple of minutes, I want to talk about the role of computer science in this process. You know, when I talk about compilers and computers and everything, one thing to keep in mind is that the idea is not, what I'm not claiming is that biological systems are like today's computers, because there are obviously massive differences. What I'm saying, though, is that computation, broadly understood, is a fundamental feature of life. These are things like decision-making, memory, integrated information processing. We have to decode the high-level control algorithms that are going to allow us to modify form and function. And after we do that, we could even perhaps build computational media that are based on these principles. So what if, and this is a hypothesis, we don't know that this is true, but what if cells are a kind of universal constructor, in the sense that what if cells know how to build towards a specified plan? And that perhaps maybe a good strategy would be to rewrite the goal state and let the cells build to the spec, instead of trying to directly micromanage the cell activities. Basically offload the computational complexity of something like building a limb or some other complex structure by, rather than trying to micromanage the individual cells, figure out how the goal state is encoded and rewrite that. And I think this is a powerful strategy, because biological systems at all scales, from chemical networks to subcellular systems to whole animals to even swarm organisms like ants, are fundamentally homeostatic, goal-seeking computational systems. If we can rewrite the goal, we might be able to achieve a lot of progress that currently eludes us. And I must say, we kind of got scooped on this idea, because this idea of goal states and micromanagement, you probably know this guy from his book, The Little Prince, but he also wrote this other book, Wisdom of the Sands, where he says, if you want to build a ship, don't get people together to collect wood and make them do all the specific things, but teach them to really drive for the goal, right? So the idea is that if the levels underneath are motivated to reach a particular outcome, you're better off controlling at that level at that top level, and that maybe this provides an efficient level of control. So for the future, what I think we at some point are going to have is a kind of biological compiler, where you can literally sit down and sort of design an animal in a particular anatomy, and you're going to exploit the modularity and the innate goal-seeking computation that living systems already know how to do, and it's going to tell you how to, it's going to fill in the steps needed to give you the creature that you want out of this. So I think that if we understood the algorithms of dynamic pattern control, basically we would have made a large progress towards resolving these fundamental issues. And of course, for engineering, we could create hybrid biological constructs with new structures and functions, new computer architectures, and so on. So I think this is really at the intersection of biology, cognitive science, and AI, artificial intelligence. So I want to thank all the people that have contributed to these projects, and our model systems who go through a lot of interesting perturbations in our lab, and of course the funders who have made this possible. And I will close by showing you some of these worms. The first time, these two-headed worms were obtained by cutting a middle fragment out of a two-headed worm. And the first time I showed this at a conference, somebody stood up and said, "That's impossible. These animals shouldn't exist." And so I like to close with this, because this is, you can't argue with the animals themselves. So thank you for listening.
Hi, great talk. How do you envision these gold states are encoded? So these are self-organizing systems, right? So the cells are interacting with each other that leads to this sort of emergent structure. Do you think it's in the rules of the rules of interaction between cells?
Yeah, so basically we're in the process now of putting together and putting out some very specific computational models that show how the self-organizing properties of these electrical circuits result in stable, large-scale shapes. So we're just beginning to understand this. We actually have some quantitative now, largely through our collaboration with Alexis in our center. This is now starting to come online where we have actual models that show how it is that these electrical patterns stabilize into large-scale stable states. And more importantly, those things can now be interrogated to ask, well, if I wanted a different pattern, what would I have to do? So we have other people working on inverting that process to say that these are the rules, but if I wanted a different shape, how would I have to modify the system? And we're going, we're using a variety of techniques, and everything from feedback training to bottom-up design to try to achieve that.
Hi, wonderful talk at the beginning. You mentioned a few times when the animal will know to stop the regrowth. And you didn't mention anything about it later. Do you have some thought about if you have a regeneration, how the animal knows when to stop the regeneration?
When to stop? Is that the question?
Yeah.
That's a great question. So my current hypothesis on this is that what tissue or organs do is they have a way to survey their own structure. So what I think they're doing is they're constantly comparing themselves to this encoded memory of what it should be. And basically the rule, if you had to write down the pseudocode for it, the rule is compare your current structure with the target morphology, and if there's still, basically it's an error minimization scheme that if there's still distance, do more. And when that error goes down to a low enough level, that's when you can stop. And you can easily imagine that there are contexts where that's going to go awry. For example, cancer. For example, there are other types of overgrowth. The system can be fooled. Okay, so it's certainly not foolproof. But I think fundamentally it's a kind of error minimization scheme where it's just trying to minimize the distance between what's currently going on and what is encoded as the ideal low energy state.
I'm just curious what you think of the role of bioelectricity in cancer, which is, I think, often thought of much more as a genetic disease, but how might that be encoded to drive tumor formation?
Yeah, so we have a couple of, and actually I took a bunch of notes to talk to Len about this. One of the projects I didn't talk about at all has to do with melanoma in our frogs. So it turns out you can, by disrupting the voltage of a particular cell population in tadpoles, you can convert all the normal melanocytes to melanoma. There's no genetic damage, there's no oncogenes, there's no carcinogen. Perfectly normal animal, you wouldn't be able to tell it apart until later on it starts to turn on a bunch of known tumor markers. And there's no primary tumor, by the way, but basically all the melanocytes go completely crazy. They proliferate, they make these crazy projections that Len just showed us. They dig into the blood vessels and the neural tube. They go into all the vasculature. I mean, it looks like melanoma. There is no genetic damage. So I'm not going to say that that's the case for all cancers, obviously, but I think it is quite possible to derange cell communication to the point where cells abandon the anatomical plan and basically go unicellular. A cell whose computational boundary shrinks to the surface of its own, itself is only the one cell, treats the rest of the organism as environment. And at the expense of the environment, you proliferate, you dump entropy, you dump waste products, whatever it's going to be, you do what you can. And so my gut feeling is that these cells, in talking electrically, but also chemically, physically, with physical forces, all of that, cells need to talk to the rest of the body to be convinced to abandon their unicellular, every man for himself lifestyle and join the body. And that process can go awry. And that process can go awry genetically. It can also go awry physiologically. The flip side of that, aside from making melanoma, which is interesting, but obviously not what anybody's looking for, the flip side of that is that, as I showed you, we can take cells misexpressing really bad, nasty KRAS mutations, and basically force them to participate in the normal morphogenesis of the body. And this has been known actually not electrically, but the fact that you can inject carcinoma cells into early mouse embryos, and they basically become part of the normal embryo. Also, regenerative systems are able to normalize cancer. That's been known for many decades, that there are patterning contexts where the patterning cues are really strong. In some cases, it can overcome what we think of as a cancer cell. So in certain cases, the genetics are crucial. In other cases, it's going to be that the cell is not irrevocably damaged, that what you have is somebody said once that if you're trying to understand a traffic jam, then understanding the combustion engine is not going to help you. It's a system level problem. And I think for a certain class of cancers, it's a systems level problem, and the system is mediated electrically. That's what I would say.
Hi. Yeah. How is the external electric field interact with the bioelectric field?
External electric fields?
Yeah,
Yeah.
we are all surrounded with that, and it seems that it doesn't bother us much. Or
Yeah, right. So it's actually pretty difficult to alter these. The gradients that I showed you are not directly altered by weak electromagnetic. What we're surrounded by is electromagnetic fields most of the time, and those people have found effects of those for sure, especially in vitro. But those don't directly alter this kind of process. The receptor mechanism for those is quite different. So that doesn't seem to be a problem for this. However, there are other aspects of the environment which are a problem. So for example, lots of drugs that are ion channel drugs, okay, for example, for arrhythmias and things like this are teratogens. They're known teratogens. They would really be a problem in embryogenesis. So I would say applied ambient electromagnetic fields are probably not a huge deal, but there are certainly drugs that you might come into contact with in pregnancy that are a big deal.
Michael, you used the word pseudocode in one of your answers, and that reminded me of one of my favorite features of the way you're thinking about this, talking about the software and not worrying about the hardware. Well, wonderful, but when I think about how hard it is to make compilers and how very, very hard it is to make compilers that can deal with something like pseudocode. I think of Charles Simone's efforts for 20 years to make what he calls intentional software without much success yet. Mm-hmm. So you're postulating another level of design that has to evolve, and I can't yet quite see what the selection pressures are that are going to help shape that level between the pseudocode and the machine.
Sure. So I would say two things. On the level of difficulty, so pseudocode would be nice. I would be happy with C at this point, right? So we don't have to go to pseudocode yet. What we for sure have to do is go beyond the machine code where we're literally pushing around individual molecules or even cells. We have to, when a regenerative process stops, or when it decides to turn a tail into a leg or whatever it is, it's measuring large-scale properties. It's measuring things that are not defined at the level of single cell. This means that somewhere this is encoded. We don't have to be able to write total pseudocode, but we have to get to the point where we understand what is this thing measuring that is not a local state. Going beyond that, selective pressure, that's a great question. And so we've actually started thinking about this in two ways. Number one, if you actually look at the tree of life and the evolution of morphological complexity, one of the things you can look at is the evolution of different types of ion channels. And some of these ion channels are incredibly interesting because some of the more advanced ion channels that appear when these anatomical innovations appear have the following property. They are not only determinants of voltage, but they are themselves voltage sensitive. What this means is that you now have a transistor, you have a voltage sensitive current. And that allows you to, so the hypothesis, and this is, we're just starting on all this, but the hypothesis is that with the appearance of those kinds of, the evolution discovered very early on, that you can make feedback loops, you can make logic circuits, you can do all these things if you have a sort of self-referential building block like that. And I think evolution found it early on. And the reason that the evolutionary pressure is going to help things like this is that this thing has to be able to remodel at a high level for the following reason. Think about the eye on the back. If every time we had an innovation in the body plan, let's say the nervous system couldn't handle it and fell apart, that kind of evolution would never get off the ground. There has to be a degree of plasticity and modularity, whereby there are circuits that make decisions about large things, like, hey, my eye is over there, I'm going to treat that as visual data. That means you have to recognize an eye. That means that it's not just a single cell kind of decision. So I think that's where the evolutionary pressure comes from. It's the evolvability and the ability to have plasticity to deal with accidents, with damage, with the vagaries of the environment. And I think evolution found this stuff very early on and has been exploited. This stuff, I mean, as you heard in the other talks, this stuff is, as bacteria have it, fungi have it. I think it came along very early on.
I think we should, in the interest of time, move to a couple other events. I'm going to thank Mike and invite Dean Glazer to come